A big hello to all NEET aspirants. In today marathon session, that is Meiosis. In earlier session we completed this Mitosis. And this Meiosis, it is important. Not only as a chapter this is important, it is interlinked with the topic that is sexual reproduction in flowering plants. And also it involves with the numericals. And as far as this Meiosis is concerned, if you keep the NCERT textbook with you, how the delivery is going to be synchronized with the text. You check it, okay? The sexual reproduction, it involves the fusion of the male and female gametes. And for the formation of the male and female gametes, a process, it is the reduction division, the meiosis. So, gametes which are invariable, it is haploid and results in the formation of the union, results in the formation of the zygote. It develops into organism. So sporophytes are formed. And sporophyte again develop into gametophyte. So sporophyte converted into gametophyte. So what type of the division occurs during the conversion of sporophyte to gametophyte? Meiosis. Where does meiosis occur in case of sporophyte? Spore mother cells. So spore mother cells undergo reduction division to produce the spores. And spores develop into gametophyte. That gametophyte produce gametes. So sporophyte and gametophyte alternate with each other. So as fertilization doubles the number of the chromosome, restored to off by means of the meiosis. And chromosome number reduced to off in meiosis, again restored by it is fertilization. So meiosis and fertilization are antithetical process, that is opposite process. Next. In meiosis, nucleus divides twice, chromosome divides once. And DNA replication occurs once just before meiosis 1. So overall, nucleus divides twice, chromosome divides once, DNA replication once just before meiosis 1. That is one point. And in meiosis, the two, meiosis 1 and meiosis 2. Meiosis 1 is purely reductional, chromosome number exactly reduced to off. Meiosis 2 is similar to that of the mitosis, it is homeotypic type of division. So meiosis 1 and it is a meiosis 2. And what happens in case of the meiosis? That is pairing of the chromosome, homologous chromosome, exchange between the chromatid parts, the recombination, the variation occur in case of the meiosis. So in meiosis, the cell in which meiosis occur, what do you call it, is meiocyte. So you might have the idea with regard to the plant kingdom. What are meiocytes in case of algae and fungi? The zygote is acting as a meiocyte in algae and fungi. What are meiocytes in case of the bryophyte? Spore mother cells. Because in case of the bryophyte, one kind of the spores, so spore mother cells are the meiocyte. And in case of pteridophyte, in case of homosporous plant, it is spore mother cell. And in case of heterosporous plant, microspore mother cell, megaspore mother cell. So trimorphic meiocytes are present in which group of plant? That is pteridophyte. And in case spermatophyte, angiosperms and gymnosperm, microspore mother cell, megaspore mother cell. So dimorphic meiocytes are present in spermatophytes as well as heterosporous pteridophyte. Monomorphic meiocytes are present in all bryophytes and pteridophytes, that is homosporous pteridophytes, like that. Next. In case of the meiosis, how many nuclei disappear? That is the 3 nuclei disappear. In meiosis 1, 1 nucleus. In meiosis 2, 2 nuclei. Total it is the 3 nuclei disappear. And number of nuclei reappear in case of the meiosis. In case of meiosis 1, 2 nuclei reappear. In meiosis 2, 4 nuclei reappear. A total of 6 nuclei reappear. So 3 nuclei disappear, 6 nuclei reappear. How many times the nuclei disappear and reappear? 2 times disappear and 2 times it is reappear, like that the questions. And in meiosis, a purely reductional, that is the meiosis 1, it is the purely reductional. And meiosis 1 is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. And prophase 1 is the lengthiest phase. And prophase 1 divided into substages based on behavior of the chromosome. Leptotin, zygotin, pachytin, diplotin, diakinesis. In early prophase 1, that is leptotin, zygotin, short living. That means short, that is the time span it is less. And in case of mid prophase 1, that is long lasting stage. Late prophase 1, that is diplotin and diakinesis. So early prophase 1, leptotin, zygotin. Mid prophase 1, that is pachytin. Late prophase 1, diplotin, diakinesis. Next, in leptotin, the word itself conveying its meaning, leptos, that is a slender threads. In case of the leptotin, maternal and paternal homologous chromosomes which are grouped, not it is a pairing, grouping at one point, what do you call bouquet stage. And visibility of the chromosome is gradual throughout the leptotin stage. Why? Because the compactation is continuous throughout the leptotin. That is, entanglement is continuous throughout what it is the leptotin. That's why visibility of the chromosome is gradual in case of the leptotin, not at a time. That is one point. And in case of the zygotin, chromosome undergoes pairing. And such a paired chromosome, what do you call it, is homologous pair. In homologous pair, length of the two chromosomes, maternal and paternal, is the same. Position of the centromere is the same. And alleles of the gene controlling the character, their positioning is also the same. Then you consider it as a homologous chromosome. Otherwise, it is a non-homologous chromosome. So, suppose these two lengths of the chromosomes, it is homologous. Suppose if you take this one, it is non-homologous chromosomes, like this. And in case of the zygotin, such a pairing, what do you call it, is synapsis. A protein complex, a protein helps to bind the two chromosomes, maternal and paternal chromosome together as a pair. What do you call this complex? Synaptonemal complex. Such a homologous pair, what do you call it, is the bivalent, otherwise what do you call tetrad. So suppose if you take the example, the maize, meiocyte, the 20 chromosomes are present. So how many bivalents are present in case of the zygotin? 10 bivalents are present. How many tetrads are present? 10 tetrads are present. In each bivalent, how many chromosomes? 2 chromosomes. How many chromatids? 4 chromatids. But these 4 chromatids are not distinct in case of the zygotin. The 4 chromatids in 2 chromosomes are very distinct in case of the next stage, that is pachytin. So 10 bivalents, 10 tetrads in case of the zygotin. And next stage it is the pachytin. Very, very important stage, long lasting stage, it is the pachytin. So tetrad stage is clearly visible. That is, each tetrad having 2 chromosomes, 4 chromatids. So in case of the maize, the diploid number of the chromosome, the meiocyte is the 20. Number of bivalents in case of the pachytin, it is the 10. Number of tetrads also it is the 10. Number of centromeres in case of the pachytin, it is 20. Number of kinetochores, each centromere having 2 kinetochore, 20 centromere having 40 kinetochores. Number of chromatids, 40 chromatids, as it is having 20 chromosomes, 40 chromatids are present. How many DNA molecule? Each chromatid can accommodate 1 DNA molecule, 40 DNA molecule. Content of the DNA, it is 4C content of the DNA. Because before meiosis 1, the chromosome undergoes what it is the duplication. Each chromosome is going to have the 2 chromatids in case of interphase. So interphase before meiosis 1 occur that results in what it is the 4C content of the DNA. So now in case of the packeting, that is 4C content of DNA. That is one point. And number of strands of the DNA in case of the packeting, as 40 DNA molecules are present, each DNA having the 2 strands, 80 DNA strands are present. 40 DNA molecule, 80 DNA strand, that is 4C content of the DNA in case of the packeting. And each tetrad having 2 chromosomes, 4 chromatid. The 2 chromatids of the same chromosome, what do you call sister chromatid? Suppose it is a maternal chromosome, 2 chromatid. It is a paternal, it is a 2 chromatid. And chromatids of the same chromosome, what do you call sister chromatid? So, it is the homologous chromosome, the 2 chromosomes, it is intrachromosomal. And it is interchromatidal, that is non-sister chromatids. It is a maternal chromosome, it is paternal chromosome. Maternal having 2 chromatid, paternal having 2 chromatid. So exchange occur between non-sister chromatids of homologous chromosome. Exchange occur between non-sister chromatids of homologous chromosome. So intrachromosomal, interchromatidal. Such exchange, what do you call it, is crossing over. At this crossing over, the recombinant nodules are formed. A spherical recombinant nodules are formed at which the crossing over occurs, that is exchange occur. And a catalyst helps in this recombination, it is recombinase. So, a new genetic recombination occur, results in variation, by selection results in case of evolution. So, very important stage in case of packeting. So, tetrad stage is clearly visible and crossing over occur between non-sister chromatids of the homologous chromosome, recombinase activity, recombinant nodules in case of packeting. Next, it is diplotin. In case of diplotin, synaptonemal complex undergo disorganization, repulsion between homologous chromosome, still united at chiasma. Here the position of the chiasma, it is intermediary. So repulsion start. It is quite antithetical to the zygote. Synopsis in case of zygote, desynopsis in case of diplotin. And still united at chiasma. Due to repulsion, the chiasma, it is X-shaped structure because of the repulsion. That is the key point. So disorganization of synaptonemal complex, repulsion, that is a desynopsis in case of the diplotin, still united at chiasma, chiasma it is X-shaped. Next, diakinesis. In case of the diakinesis, chiasma move to the tips of the chromosome like this. Earlier like this, now it is like this. Tips of the chromosome. So, still it is moving. What do you call terminalization? Movement of chiasma to the poles of the chromosome, what do you call terminalization? And maximum condensation occur in case of diakinesis. That is one point. And is a transition to metaphase 1. And meiotic spindle assembled to separate this homologous chromosome, not separated. To separate this homologous chromosome, meiotic spindle assembled in case of diakinesis. So diakinesis is a transition to the metaphase 1, and as bivalent migrate to the periphery of the nuclear membrane disorganizes. So maximum condensation, nuclear membrane disorganizes, terminalization in case of diakinesis. Next it is metaphase 1. In metaphase 1, the bivalents which are arranged at the equator. So it is maternal chromosome, it is paternal chromosome, it is the point of the contact. And telomeric ends of chiasma are united. This is the polarized end. So telomeric ends of the chiasma united at, it is metaphase 1. So bivalents at equator. Centromere directing towards what it is the poles. So how many bivalents are present in case of metaphase 1? As you took the example of this maize, the 20 chromosome. So 10 bivalents at equator. How many chromosomes? 20 chromosomes. How many chromatid? 40 chromatid. That is 20 maternal, 20 it is paternal chromatid. 10 maternal chromosome, 10 paternal chromosome. So, telomeric ends of chiasmata are united at metaphase 1. Next, anaphase 1. So, it is the bivalent. It is the homologs. So, homologs in bivalent got separated. And each chromosome having how many chromatid? 2 chromatid. So, separation of maternal and paternal in case of anaphase 1, what do you call disjunction? Bivalents are not disjuncted. Homologs in bivalent got disjuncted. And each chromosome having 2 chromatid. So, in this pole it is the 10 chromosome, in this pole it is the 10 chromosome. So, 2 poles in a cell it is the 20 chromosome. But chromosome number exactly reduced to off. At each pole it is a 10 and another pole it is the 10. At each pole it is having 20 chromatid. In this pole it is having 20 chromatid. In this pole it is the 2C content of DNA. This pole it is the 2C content of DNA. In a cell it is the 4C content of the DNA. So content of DNA at each pole in metaphase 1 cell is 2C content of the DNA. Number of chromosomes at each pole in case of anaphase 1 cell, it is 10 chromosome. Number of chromosomes at each pole in this, it is the 10 chromosome, and chromatid it is going to be 20 chromatid. So 2C content of the DNA in case of anaphase 1. So ploidy of the gamete decided at, it is anaphase 1. Disjunction in case of anaphase 1. Next it is the telophase 1. Whatever the chromosome reached to the poles undergo despiridization, decondensation, reappearance of the nuclear membrane. So, in a cell, number of nuclei formed, it is the 2 nuclei, and each chromosome with 2 chromatid. In this telophase 1, chromosome do undergo some dispersion, but not extremely at extended state of interphase. What is the meaning in that one? In case of interphase, each chromosome having only 1 chromatid. So dispersion it is the more, but it is having the 2 chromatid. That's why chromosome do undergo some dispersion, but not extremely at extended state of interphase, as it is having 2 chromatid. And it is followed by cytokinesis, results in dyad of cells. So in each cell, the content of the DNA is the 2C content of the DNA. Number of chromosome 10 and 10 chromosome. Number of chromatid per cell 10 into 2, that is the 20 chromatid. In another cell it is the 20 chromatid. The 2 cells are formed. So dyad of these cells at the end of telophase 1. After meiosis 1, before meiosis 2, there is an interkinesis. It is similar to the interphase. But interkinesis is of short duration. Why interkinesis is of short duration? Because in case of interphase it is having G1, S and G2. But in this interkinesis it is having G1 and G2, but not it is S. Why S stage is not required in this? Because already the chromosome having 2 chromatids. So there is no requirement of for it is S stage. That's why interkinesis is of short duration. Already chromosome number got reduced. But why there is a necessary of the meiosis 2? Because each chromosome having 2 chromatid, the chromatid has to distribute it to daughter nucleus. One chromatid has to be distributed to daughter nuclei. So it is inevitable that meiosis 2 it is must. So as to distribute one of the chromatid toward its nucleus. So interkinesis short duration, meiosis 2 it is must. Just before meiosis 2, that means the interkinesis, and before the chromosome fully elongated, that is immediately after cytokinesis, before the chromosome fully elongated, the meiosis 2 it is initiated. In meiosis 2, prophase 2, metaphase 2, anaphase 2, and it is the telophase 2. And in case of the prophase 2, that is nuclear membrane disappears, the chromosomes are arranged in the 2 cells, as similar to that of the prophase. And in case of the metaphase 2, 2 spindle apparatus form at right angle to the spindle apparatus of what it is metaphase 1. In case of metaphase 1 like this, now it is like this. So 2 spindle apparatus formed in case of metaphase 2. So earlier in case of metaphase 1, 1. A total of 3 spindle apparatus formed in case of meiosis. In meiosis 1, 1. In meiosis 2, 2. That too in case of metaphase 2. So chromosomes are arranged at the equator. We are not calling it the bivalent. Chromosomes are arranged at the equator. This is the one cell, this is the one cell. In one cell, chromosomes are arranged at equator. In another cell, chromosomes are arranged at equator. So in each cell, chromosomes are arranged at the equator and at equatorial plate. So here the 10 chromosomes, 20 chromatid, 20 chromatid. Here 2C content of DNA, here 2C content of the DNA in case of metaphase 2. And in case of anaphase 2, suppose in case this is one cell, the chromosomes are arranged like this. So in case of anaphase 2, splitting of the centromere, one splitting of the centromere, chromosomes are pulled to the respective poles of each cell. Now, earlier it is having 20 chromatid in a cell. Now it is in each pole 10 chromatid, 10 chromatid. 10, that means in this pole it is 10 chromosome, this pole 10 chromosome. So each pole having what it is 1 chromosome, 1 chromatid. So 10 chromosomes, 10 chromatid. 10 chromosomes, 10 chromatid. So 1C content, here 1C. In another cell, like that, 10 chromosomes, 10 chromosomes, 1C content, 1C content. So, splitting of the centromere occur, each chromosome is going to have 1 chromatid. So, what is the difference between the chromosome of anaphase 1 and anaphase 2? In anaphase 1, each chromosome having 2 chromatids. But in case of anaphase 2, each chromosome having 1 chromatid. Suppose it is the cell, it is one pole, it is one pole. 1C content, 1C content. If there is another cell, 1C content, 1C content. So 10 chromosome, 10 chromosome, 10 chromosome, 10 chromosome. So chromosome number doubled in case of anaphase 2 cell. And telophase 2, reappearance of nuclear membrane. So 4 nuclei which are reorganized. So number of nuclear membranes reappear, the 4 nuclear membranes reappear in case of telophase 2. And followed by cytokinesis results in what it is tetrad of the cell, 1, 2, 3, 4 cell. Each cell having 10 chromosome. In tetrad of the cell, sum of the chromosome present in the tetrad of the cell is going to be 40. Each cell, haploid cell is going to have what it is the 10 chromosome. The content of the DNA, 1/4 of the content of the DNA transferred to the each haploid cell, that is 1C content. So out of the 4 cell, 1C, 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 1C. In case of telophase 1, that is the 2 cell, 2C, 2C. Now it is the 4 cells are formed. Each cell it is 1C content of the DNA. Earlier it is the 4C content of the DNA. Before meiosis 1, now it is the haploid cells having 1C content of the DNA. So the haploid content, that is 1C content of what it is the DNA. That is one point. And what is the significance of meiosis? So chromosome number kept constant from generation to the generation by means of what it is meiosis. And this meiosis per se paradoxically, that means by itself, surprisingly, reducing the chromosome number to half. So what is the paradoxical event in case of the meiosis? Chromosome number reduced to half. Not only a paradoxical event, chromosome number reduced to half, it is also responsible for creation of genetic variation across many generations. That is the meiosis. And in next video, just we'll go through this what it is, that is the questions. Whatever the things, knots and the twist present in case of the text, we will go through these the textual questions, that is based on what it is the NEET and the previous questions also. Okay, thank you very much.